Okay, so we might make a start. Welcome to the 2024 Chalmers Oration. This is a very important event in the life of the university and the College of Medicine and Public Health. It's so great to have you all both in the room and also online. Before we start, we need to acknowledge critically our local traditional custodians of the land, and to that end I'd like to welcome Petiola Wilson to the stage, Prangana Narangeri Nadjeri Man. Thank you, Petiola.
Yakiari yariangori, yakiari yariango, yakiari yariango. Doregone rena so, doregone rena so, doregone rena so. Kaniyata. So we say Yugari Yakara, Marn Pundi, Dalamanisha, Kaniyata, Nari Petiola Wilson, Narandri Kana and Ranga, Tongan Mirna. Nawangandi. Nawangandi means what I am saying is, brothers and sisters, I meet and greet you on the lands of the Kana people. My grandfather's country. When I say Nainari, I say my name is Petiola Wilson, and I represent Narandri Kana and the Ranga and the Tongan communities. So it's important for me to always still pay respect to my Tongan heritage, because it's not just being an Aboriginal person, so that makes me who I am. Grateful to be here today. Thank you to Latitia and Flinders University Takatina for having me here, which means family. As soon as I knew it was Georgina, and I was lucky enough to go to Australian of the Year conference at the Convention Centre and listen to Georgina and Richard speak, and I've been going to that place for like 15 years on big events. I don't even listen, I just go there for the food. But this time I actually did listen to what he was saying. And yeah, I really did. I knew bits and pieces about it and whatnot, but I didn't actually, no one actually really catches my attention, because I'm like off wondering about everything else like a lot of us are doing. So I actually sat there through that dinner and was like, yeah, actually interested by the end of it. I was like, realised what you do and what you have done. So we come here from all parts of Australia and from around the world. So we say Manda Pundi Kana Yaratana, which means we're glad you came to Kana country. Keep your culture, share your culture, embrace our culture, and continue to grow together. And then we say Flinders Takatina Marachanga Kana Yaratana, which means Flinders family. On behalf of the Kana people, we say welcome to you and your families, and let's all become one. So Natalia, thank you. I'm glad I come out and done this. I'll see you at your awards night and everything next week, but I have to leave now and go pick my daughter up from school. So a lot of you are like, yeah, and she would sit there and listen to you as well. But even on the ABC channels and everything, when they show the stories, it does really, doesn't make us, yeah, it's big words, because we don't understand a lot of the words that you're using, but the scientists in the room will, and you still can captivate our audience of the less educated ones. That's what I like to say. But yeah, so Natalia, thank yous, and please enjoy today, and just always remember it's Takatina Kuma, which means Takatina first, and always worry about your families as well as I do. So Natalia, thank you, and enjoy. Like, you will get captivated in this. You should have grabbed more cookies and filled your cup as well. So thank you.
Thank you, Petiola. And this is for those of you who are new to this building, which I think you'd agree is the most extraordinary structure, both in terms of its functionality, like this is state-of-the-art health and medical facilities, but also there is this nobility. This building sits beautifully on this land, and in part, in fact, very much part, because Uncle Yellowburka Lewis O'Brien and others provided their cultural advice and counsel. So what you'll see across all of the floors is different colours of the rainbow serpent and rainbow sand. So this infuses this deep sense of how noble and how important the work is that happens here. So I too want to acknowledge that we're on Kaurna land. Thank you, Petiola, for your welcome to country, and also acknowledge that Flinders has the privilege of working across many lands across South Australia and into the Territory, and I want to acknowledge all the traditional custodians of those lands. So Georgina who is our 2024 Chalmers Orator. I'll introduce her in a minute, but she is happy to take questions both in the room and online. And for those particularly online, if you want to provide your questions, you can do so via that Slido QR code. So we're here to acknowledge the foundation work of John Chalmers AC. John and his wife Alex are here in the room, and so it is so good, John and Alex, to have you both here and family. So John was the first Professor of Medicine for over 20 years at the Flinders Medical Centre at the College of Medicine and Public Health, and in many ways provided that foundation of the criticality of research in an environment of education. This year is also particularly important because it's the 50th year that we've celebrated of the medical program at Flinders. We had an event not so long ago which we also want to recognise the other foundation professors, and I think Warren Jones, AO, you're in the room, a Professor of Obstetrics and Gynecology, and we want to acknowledge the extraordinary legacy that these people played for the work that we are doing here in this building and across the medical school. So let me introduce you to Professor Georgina Long, AO. Her CV is very long and we don't have time to go through it all, but in a nutshell, she's co-director of the Melanoma Institute Australia and chair of Melanoma Medical Oncology and Translation Research at the Melanoma Institute and North Shore Hospital. She leads an extensive clinical trials team and laboratory at that institute with a focus on targeting therapies and immuno-oncology in melanoma. She is the principal investigator on a number of clinical trials aimed at improving the lives and outcomes for people with metastatic melanoma in particular. She has many awards under her belt, to name a few. Appointed Officer of the Order of Australia in 2020, recognised with the Australian Academy of Health and Medical Sciences Outstanding Female Excellence Research Medal in 2021. 2021, the prestigious Ramaciotti Medal for Biomedical Research. 2023, the European Society for Medical Oncology Women for Oncology Award. 2024, Fulbright US Mission Australia Award for Leadership Excellence, and 2024, must be hot off the press, Fellow of the Australian Academy of Science. And this year, as you would all be aware, in recognition of her groundbreaking research and work, she was named Australian of the Year. So please join me to welcome Professor Georgina Long to deliver the 2024 Chalmers Oration.
Thank you, Jonathan, for that incredibly warm introduction. I'm just going to shift a few things on the podium so that I can talk to you all. So, I'd also like to thank Flinders University for such gracious hospitality today. I'd also like to acknowledge the traditional custodians of the Adelaide region, the Kaurna people, and their elders past and present. But importantly, I want to reflect on how much we really know about our First Nations people and the land that they've nurtured for 65,000 years. What can we learn from them regarding sustainability, given their deep respect for the land and the waters and sky? I'm truly honoured to be here to deliver this oration, and I'd like to acknowledge Professor John Chalmers AC, his wife Alex, and family. But what is most evident to me is the great love for Professor Chalmers as a mentor and a source of inspiration for many. A clever human and a remarkable legacy from his dedication to science, and commitment to supporting young researchers. I've had good reason to reflect on clever people and the power of their legacy, because I lost my dad, Dr. Geoffrey Long, in September. Dad was an anaesthetist who pioneered the availability of epidural anaesthesia in labour wards in the 1960s. Amen for him, I hear mothers whisper under their breath. At home, my dad was an amateur botanist who understood the planet's climate problems early, alarmed by the scientific implications in the evidence from the Antarctic cores. He was an avid reader, and I grew up with an extensive and very broad home library with rare books, a huge range of fiction, poetry and plays, but a massive collection of non-fiction, medical text, history and art books. Dad had a philosophy of keep moving forward and keep your goals in mind, always about improving things for humankind, especially when things get tough. He really understood that life has very tough parts for all of us. It was part of the human condition. And therefore, we had to develop ways to manage that reality. He invented the Forgettory, just for me. He would say to me, put it in the Forgettory and move on. Life is short, so make the most of it. I'm reminded of my dad through the reflections of author Maya Angelou. I want to be remembered as the person who contributed to the legacy of the world, who did the best I could to help those in need, she said. Today, I've been asked to reflect on curing cancer, and particularly my personal journey. Well, unfortunately, we certainly have not cured all of cancer, and I'm very much still on that journey. Don't get me wrong, there have been some incredible milestones in my career, but I believe our lasting legacies grow as we grow. They grow in us, they grow beside us, they grow from us. Some legacies develop through integrity, making good choices and creating a positive impact. Some legacies reflect the choices we've made or the situations we've been in and the actions we took and the consequences that those had. Some legacies are unfair, unimaginable, terminal. Some are hard-earned or fought for, some are fate or even accident. Often, legacies are a combination of all these things in one way or another. For those of us in science, we understand it is the impact of what we leave behind for those who follow after us. And we understand that because of those who have gone before. So on my journey, where to begin? Probably at the start. My parents were doctors and researchers. Dad, an anaesthetist, as I said, and Mum, Anne, a university academic and a doctor in public health. They had 6 children. This will sound like a familiar tale for Professor Chalmers, who I believe has 5 children of his own, while he and his wife juggled their medical careers. I was the second youngest, so I needed to have a bit of grit. My mum was about thinking big, always talking about actions that can impact populations, clean water, sewerage systems, sanitation, vaccination programs. These all impact population health and productivity. Not just for one, but for all. How can we get this out to more? How can we get it out quickly? Her mantra was make a difference to many. Dad was planted in reality. He had no time for drama. Although he never used that word, it was in his actions that I knew he had no time for drama. His favourite mantra was, "You can never get enough education." All 6 kids remember this mantra well. We talked about everything at our dinner table, and being the second youngest of 6, you can imagine my life education started very early. After what I've heard over Sunday roast, including a highly medicalised sex education, it's amazing that I have had any children at all. My parents were adventurous and curious people, and I live by their mantras. When I was little, we lived in the US for about a year and a bit. Mrs. Black was my year 1 teacher in Philadelphia. She kept me back one afternoon until I learned how to find words in a dictionary, because everyone else in the class could, except me. Looking back, this was at a very early age. I was only just 6 years old. And I remember it was the first time that I realised that I can do things if I apply myself. Nothing is really too hard. I had that aha moment of getting the hang of something, of completely understanding what was required, what was necessary for me to achieve. I had a new lens to view the world. From there, the aha moments kept coming. I loved them so much. I sought them out. I completed my PhD in chemistry under the supervision of an incredible woman, Professor Margaret Harding, in the Department of Chemistry at the University of Sydney. I examined how an anti-cancer agent, streptonogrin, bound to DNA. I was then back in the US for postdoctoral studies at Scripps Biomedical Research Institute in California after receiving a fellowship, a Fulbright fellowship. I worked on nanotubules. My Fulbright experience taught me that in life-defining events, we don't just have a responsibility, but an obligation to ensure our vision is groundbreaking. And at Scripps, the expectations for achievement were extra. In fact, extra was normalised. It was even expected. It became the norm for me, and it reinforced an incredible work ethic. Research had to be so impactful that it would be worthy of nothing but the front cover of Nature or Science. I was taught, yes, there is huge possible failure, but there is also huge possible success. In fact, failure is a badge of honour because you tried. I'm just turning off my phone because it keeps going crazy. And the only way to deal with it is to turn it completely off. So, failure as a badge of honour. And aha, I realised that the extraordinary is only achieved when we think big, we think outside the box, and we take big risks. When you do things like a Fulbright fellowship, and you go overseas and you study, you learn through different lenses, which give you valuable perspectives. And what I loved most about my American experience was the celebration of clever people and clever ideas. The US environment has historically been about accelerating opportunity and embracing innovation, fostering excellence, and generating change in a highly efficient and productive way. My American experiences taught me other things too: optimism, confidence, ambition. These attributes are celebrated. They're not always celebrated in women, but they have given me important cut through throughout my whole career. And in return, I learned to celebrate clever people and clever ideas. There is nothing better than an idea. And even ideas you may think are not very clever can start a discussion that is important and big. Innovation and creativity, thinking outside the box, it's not only my passion, it drives my expertise. So because I grew up with medical books that my parents owned, after completing my PhD in postdoctoral studies, I felt it could be supplemented with an in-depth medical knowledge via a degree. So I completed a medical degree. I've often described the next part of my journey beginning with sliding door moments. During my medical degree, I earned money taking blood and inserting cannulas at Royal Prince Alfred Hospital on weekends. I also helped one of my favourite lecturers, Dr. Kerry Crotty, organise her office. I got the job with her having done a project on melanoma during medical school. It was a project on melanoma of the head and neck. You see, she reported pathology cases for the dermatologists and the surgeons at Sydney Melanoma Unit, which later became Melanoma Institute Australia. During that time, I had a chance meeting with a pathology registrar, a pathology trainee called Richard Scollier. Kerry Crotty, Richard Scollier, and my paths crisscrossed over several years. Kerry added dermatology to her training and Richard became a consultant pathologist, looking down the microscope, looking at parts of human tissue to confirm a clinician's suspicions, or not. The clinician would send the biopsy, abnormal skin lesion, for example. Is this melanoma? Is this a complex nevus or mole? I completed my medical degree. I married my best friend, Greg, my greatest supporter, my sounding board, who completely understood unconditional love and commitment, completed my junior doctor training years, and finished my specialty training in medical oncology. Drugs and chemistry. You cannot get better than that. My passion. I had just had my third baby. Yes, I had 3 kids during my specialty training and they did not pop out like that. And I was considering which area of cancer to specialise in, having now got my letters as a specialist. Richard Scollier, was cycling past my backyard in Sydney and stopped to chat. I had the idea of specialising in melanoma. Why? Because it was the least sexy cancer for a medical oncologist to do. There were no drug therapies that prolonged survival, and I saw an opportunity. We had so much melanoma in Australia. Something had to give. Just a short year prior, I had completed one of my training terms with Dr. Anne Hamilton, the melanoma medical oncologist at Royal Prince Alfred Hospital. I was struck by how hopeless it was for patients where the melanoma had spread, stage 4 or advanced melanoma. We had nothing. It was hard. And you will recall I had done a melanoma project with Dr. Crotty, so was very well aware of the statistics. Richard, in the backyard, was excited and encouraging me to follow my instincts. You're crazy if you don't get into melanoma, he said. Reach out to Rick Kefford, he advised, a medical oncologist at Westmead who saw a lot of melanoma and attended our weekly multidisciplinary team meeting at the Sydney Melanoma Unit. Melanoma and its risks were not new to me. Even before I completed medicine, my father would warn us of the risk of damage from ultraviolet rays and would wear long sleeve shirts and wide brim hats outdoors at a time when sunburns weren't thought of as seriously as they are today. There were many opportunities to perform research and to make a difference in melanoma. My first 4 years as a medical oncologist and translational researcher were self-funded by grants I wrote. At that time, despite having 3 children, the oldest of whom was only 4, I was not allowed to take the grant part-time. This was challenging as I had to juggle schedules at home, the clinic, and the research lab. Fast forward more than a decade, and in 2017, Richard and I become the co-medical directors of Melanoma Institute Australia. The melanoma drug treatments I developed in humans, along with leading melanoma medical oncology colleagues around the world, have broken new ground, and many thousands of lives are saved as a result. I'm going to stop here for a minute and describe this slide. On the left are Adam and Josh, and their two children, Grace Georgina and Mia, MIA, Melanoma Institute Australia. Those men, this is a photo taken many years after I had started them on a clinical trial of immunotherapy. They are alive and well today, more than 10 years after starting immunotherapy, off treatment with no signs of advanced melanoma, both starting with stage 4 melanoma. They now have, those two children on their laps are now 5 and 6. On the right is Bert. He's 102. At 101, he was given 1 or 2 doses of a single immunotherapy drug because he had a very symptomatic melanoma at his neck. Complete response. He lived to 105, did not die of melanoma independently at home. It's a true game changer. So when did this all start? This all started in 2009 when my mentor and colleague, Rick Kefford, who you recall I mentioned earlier, from Westmead, and I started our first phase 1 clinical trial, a first-in-humans trial of BRAF-targeted therapy. At that time, advanced melanoma was an almost certain death sentence within months. It was the incurable cancer, the cancer you did not want to get. I then shifted my attention to immunotherapy. These drugs have revolutionised not only the survival rates in melanoma, but many other cancers. But more of that in a minute. Mid-2022, Richard and I addressed the National Press Club of Australia. Our aim was to put melanoma prevention in the national spotlight. Prevention is better than a cure, and I know that's a cliche, but for every dollar spent on primary prevention of melanoma and skin cancer, the health budget gets up to $10 back. We called for more to be done to promote sun safety across Australia in all age groups and in sport. And we pleaded with social media and advertisers to stop glamorisation of tanning. The response was instant and impactful. Social media giant TikTok was spurred into action, launching its aptly named Tanning That's Cooked campaign, a first for a social media powerhouse. What a public health achievement was that. Government and policymakers began working closely with us on several initiatives, including to improve sun safety in sport, which we launched in December 2023 at the Australian Institute of Sport. We were also delighted to join Prime Minister along with Education and Health Minister to launch the Parliamentary Friends of Melanoma at Parliament House. Australia does have the highest incidence in the world. Someone is diagnosed with melanoma every 30 minutes, and someone dies from melanoma every 6 hours. I saw the challenge of melanoma as a tremendous opportunity to make a difference to many. Key to shifting the dial has been Melanoma Institute Australia's model of embedding research into routine clinical care. So this is how it works. Questions arise in our clinics. These questions then inform our research priorities and program, which underpin new research programs and clinical trials. And we then apply these results back into our clinics, into our guidelines, and we implement them to benefit patients. This is, again, a cliche, bedside to bench and back to bedside, or otherwise known as translational research model, but it is real. We take a multidisciplinary approach, all working towards our goal of 0 deaths from melanoma by contributing to groundbreaking clinical and research outcomes. For example, that some beds cause melanoma, that less is more for melanoma surgery, that whole brain radiotherapy does not help melanoma patients when the melanoma has spread to the brain, that the prognosis of early melanoma is driven by specific pathological features. So back to my area, medical oncology, drug development, chemistry, immunotherapy in cancer. Immunotherapy or checkpoint inhibitors work by unleashing the body's immune system to destroy the cancer cells. Immunotherapy has been a transformative revolution in cancer management. So I'll just explain this scan here. On the left, 2018, a patient with all those black areas throughout the bones, metastatic melanoma. Her survival would have been weeks. On the right is a recent scan. She's alive and well today, and in fact, yesterday morning in Sydney, I swam with her the Coogee Island Challenge. She's doing very, very well. But what you may not realise is that immunotherapy was pioneered in melanoma first, and other cancers followed, for example, lung, kidney, triple-negative breast cancer. And Melanoma Institute Australia remains at the forefront of global immunotherapy clinical trials. Now, more than 50% of advanced melanoma patients are cured, a term the cancer field does not use lightly. I now use immunotherapy for earlier stage melanoma, so before disease has spread around the body. But as you well know, advances like these are built on decades of research, incremental failures, incremental successes, and the legacies of others. I am proud of what we've achieved in melanoma research, but at the same time, I acknowledge the many families where these breakthroughs came too late. At Melanoma Institute, we are forever indebted to those loved ones and all the patients for their selfless commitment to research, which has changed the future for others. It is Aussie mateship at its best. Today, the Institute is pushing the boundaries further to cure even more people with melanoma. Earlier this year in Chicago, at the largest and most globally prestigious oncology conference, the American Society of Clinical Oncology, also known as ASCO, we announced an astonishing breakthrough from the Phase 3 NADINA trial. The NADINA trial was designed by me and my colleague, Professor Christian Blank from the Netherlands Cancer Institute back in 2018. We have a long-time collaboration. With our multidisciplinary approach, we showed that giving combination immunotherapy before surgery in high-risk early melanoma, that's called neoadjuvant therapy, versus the current standard of surgery, then drug therapy, immunotherapy, called adjuvant, we reduced the risk of recurrence or death from melanoma by a further whopping 68%. It is a simple concept. The immune system is better able to see the enemy and be trained against it in a timely manner, and thus more effectively mop up any cancer cells that we cannot see. I liken it to sniffer dogs being trained through exposure to illicit drugs so that when they get to work, they know what they're searching for. We can now confidently say that giving immunotherapy before surgery, neoadjuvant, is better at preventing melanoma from spreading than giving it after surgery. And furthermore, 60% of patients did not require any other treatment after 2 doses of immunotherapy before. This is an absolute game changer. Then something awful happened. In May last year, Richard, my friend and colleague, was diagnosed with a grade 4 IDH wild-type unmethylated glioblastoma. It sounds bad because it is bad. It is the worst subtype of brain cancer, a brain cancer of which the standard treatment and survival rates haven't changed in 20 years, and that's not for want of trying. It essentially is incurable. Few people survive it, and it nearly always comes back. With the standard treatment, which is surgery to remove as much of the tumour as possible without causing any major deficits, followed by an intense course of radiotherapy, maximum dosing of radiotherapy, 60 gray, and chemotherapy, survival still remains quite short. The majority of people die within 24 months. I have known Richard for more than 25 years, and his glioblastoma diagnosis was devastating. He was 57 years old. I have put thousands of patients on immunotherapy. I have managed their side effects. I have monitored their progress. I have designed, written, and led multiple immunotherapy clinical trials, including melanoma that spreads to the brain. I knew that if anything was going to have an impact or might have an impact, it could be immunotherapy. Those days, those early days after Richard's diagnosis, were spent researching glioblastoma. I hadn't done it since my training. I spent hours with international glioblastoma experts who generously gave me their time, and pharmaceutical companies. It was the week leading to that world premiere oncology conference I told you about before, ASCO. I never miss it. I almost cancelled, but then decided I'll go and utilise every available moment between melanoma and glioblastoma to try and understand the drug landscape for glioblastoma. I attended many glioma sessions as I could, and I met with experts in neuro-oncology from across the globe. I trawled through the scientific literature and medical journals, searching for the latest papers and treatment options. It was pretty barren. You could count the number of immunotherapy clinical trials for glioblastoma on one hand. They'd all failed. After all that work, and despite the presence of a passionate group of clinician researchers and scientists within the glioma field, there was little interest in drug development in glioblastoma from pharmaceutical companies, particularly for Richard's subtype. I had not experienced that resistance before. In an absolute travesty, there was not one single clinical trial available for Richard, and that is upfront, first line, not one. Perhaps I was naive in my expectation. I've been told that before. In melanoma, every patient, particularly those with the most difficult to treat subtypes, and remember, 45% of melanoma patients are still dying, are considered for clinical trials. And I have many trials open at any one time. So I was disappointed and frustrated. So I had an idea. As scientists and researchers, it is incumbent upon us to push the medical field forward safely. Melanoma had broken new ground, and many thousands of lives had been saved as a result. Could any of my learnings be applied to the most difficult to treat cancers such as glioblastoma? I devised a plan to treat with neoadjuvant multiple checkpoint inhibitor immunotherapy and a personalised vaccine. But I then had to implement that plan and ensure that the scientific analyses of the tissue and blood were done. Dr. Brinda Shivalingam, the neurosurgeon I had worked with in melanoma for over a decade, was also a specialist in glioblastoma surgery, and she exclaimed, "Georgina, I have been waiting 10 years for an approach like this," referring to the neoadjuvant combination immunotherapy plan that I'd come up with. Richard arrived back from Poland, where the tumour was discovered after a seizure. He was sad. He was scared. He knew what was ahead of him, and he could hardly think past the minute he was in. When I presented my idea, the ray of hope I had given him was palpable. I see this all the time in my melanoma patients. Richard came alive. His spirits lifted. Consenting to the novel drug therapy plan was the easy part for him. As he always puns, it's a no-brainer. It was consenting to the standard aspects that Richard found a challenge. How much surgery to have? How much radiotherapy? What dose? Could I have less than standard? In the end, Richard had standard surgery, delayed only 12 days for the neoadjuvant combination immunotherapy, standard radiotherapy, and then adjuvant immunotherapy for 12 months, the standard adjuvant time course for immunotherapy, along with a concurrent personalised vaccine. My colleague, Professor Helen Rizos, an incredible scientist, cell biologist, and chair of the Melanoma Institute's research committee, shared my vision that we had an opportunity, actually an obligation, to try something groundbreaking, but within the limits of safety and ethics. She and a peloton of amazing women gave me the strength to implement my plan. Just like a bike race, together heading forward, protecting one another from the headwinds, but having to do things in no time. Helen and I gathered the Melanoma Institute's translational research teams, head of Biobank, Nick, she's there at the left, very left photo at the corner there, and mentee, now colleague, Dr. Inesh De Silva, she's at the left-hand photo at the right. Helen Rizos is on the computer screen. This is the Tuesday before Richard's even arrived in the country. And we're planning the scientific analysis. We began strategizing about the experiments. We invited the neuropathology team and others from around Australia. I turned to my own medical oncology team to implement the clinical treatment plan. Maria Gonzalez, top right, Director of Clinical Care and Clinical Trials, helped me apply to the authorities. I submitted the paperwork as the physician who created the plan with the help of Kate Willis, bottom right, the clinical nurse consultant. Together, we look after melanoma patients. She is an expert in immunotherapy care. I met with 20 biotech and pharma companies at all hours of the night. Helen, Inesh, Maria often joined me. I designed the vaccine with Helen Rizos. Maria Gonzalez helped me import it, my good colleague and a medical oncologist who has walked the melanoma journey with me for 15 years. He was my PhD student years ago. Associate Professor Alex Menzies agreed to support me along with Dr. Inesh De Silva and Dr. Matteo Carlino, if ever needed. We are not brain cancer specialists. This was a new field for us, but it takes an inspired team. Helen Rizos and I were proud to give hope, proud to lead the effort, conceiving the plan, ensuring clinical risks were mitigated, as I do in my clinical trials in melanoma, and Helen Rizos driving the tissue analyses. We know how melanoma works. We know how cancer works. And we know how immunotherapy works. There was natural pushback, as there should be when pushing forward with novel ideas. This cancer is in the most precious and critical organ, the brain, and that organ is contained within a closed box, the skull. There were no clinical trials, no protocols for using immunotherapy in this way for this type of brain cancer. And swelling could be exacerbated by immunotherapy with dire consequences. And with that came a lot of fear, because you cannot live without a brain. But the upside of immunotherapy is that it may deal with this problem by stimulating immune cells to selectively kill cancer cells and preserve the normal brain tissue. Brain swelling was only a relative risk and likely low from my experience in melanoma that had spread to the brain. Other barriers were put forward, that the brain cancer is immunosuppressive, so it would not respond to immunotherapy, and that brain cancer is heterogeneous, meaning full of different cancer cells, so impossible to target all of them effectively. But we have those problems in melanoma too. These weren't barriers for the peloton. I had navigated all of these barriers in melanoma, including using combination immunotherapy to treat tumours which had spread to the brain. Of course, we had fear. It didn't stop us. We took very calculated risks together. We inherently understood the risks that may extend or shorten Richard's life. Richard may survive and beat the unbeatable. At worst, he would leave a legacy, and we will have increased scientific knowledge for the benefit of future brain cancer patients. So combination immunotherapy, true neoadjuvant, had never been used in brain cancer. Frontline, first up, before surgery, before radiotherapy, before chemotherapy. As I mentioned earlier, in melanoma, we have proven that it is much more effective to give immunotherapy just like this, before definitive surgery, so that the immune system is more activated to destroy the cancer. We'd proven it in the Nadina trial. In brain cancer, there have only been a very small number of international clinical trials using immunotherapy, but none using combination frontline before surgery. So we did it, combination neoadjuvant immunotherapy up front. Our scientific analyses on the brain tumour removed 12 days showed, firstly, there was a 10-fold increase in immune cells within the tumour. Secondly, these immune cells were activated against an enemy. And thirdly, the immune cells within the tumour were bound to the drug therapy, providing something we'd already shown in melanoma, that there is no blood-brain barrier as historically conceptualised, preventing the drugs from reaching the tumour. We could not have hoped for better results. It was doing what we'd seen it do in melanoma, but this time an inkling in glioblastoma. Melanoma Institute and the melanoma field has led the world in the development of personalised cancer vaccines. Using the scientific data from the DNA and RNA from the glioblastoma, Helen Rizos, Ishmael Vergara, a senior scientist in our lab, and I collaborated with a tiny biotech I worked with in melanoma to develop the approach to a personalised vaccine to further boost the immune system against the glioblastoma cancer cells. Helen and I designed the final vaccine. We are now looking at the laboratory data analysed by Helen to determine the effectiveness of the vaccine. Now, caution. While this early scientific evidence is promising, this is only N of 1. And although Richard is without recurrence so far, this is not evidence. Evidence must come in the form of a clinical trial. And this is how good science is done. That is how 10 drugs for melanoma are now available on the PBS, funded for all Australians because of clinical trials, including many investigator-led trials, demonstrating a significant benefit to patients with melanoma. The survival of patients with melanoma, once 25% at 1 year, is now well over 50% at 5 years because of clinical trials. In fact, I started planning the clinical trial in glioblastoma within weeks of Richard's diagnosis. That is how I approached melanoma. By September 2023, I secured the drugs and had the basic trial design. I handed this over to a committed and driven young and up-and-coming clinician researcher at Peter Mac and at WeHi, Dr. Jim Whittle. He was the final architect of the protocol, and he had it completed in record time with a colleague from the USA. It takes time to open a human experiment, a clinical trial. Good scientific conduct requires the right checks and balances. What if we did the trial and found it worked only in 10% of people, but 50% died of toxicity? This is why clinical trial work, from idea to implementation, takes time. As a scientist, it is ingrained in me to treat these initial results with the utmost of caution. We still do not know whether the immunotherapy has made an impact. 5% of patients with glioblastoma can survive more than 3 years on the standard therapy. Richard may be one of those. What was achieved, though, with resistance from some, but support from a peloton of scientific women, was to show that in some patients it is feasible to give drug before surgery, and therefore measure the impact of novel drugs on the cancer. It's also going to take a global effort to test whether the promising results from one patient can make a difference to many. But as the leader of this research in glioblastoma, I am hyper-vigilant about our team's collective responsibility to ensure pure scientific process is adhered to. It would be easy for us to get carried away with this incredible opportunity our research is creating, but we must follow the process and ensure we're not jeopardising this for future people. For us in the cancer field, courage is needed to push boundaries, backed by science for the benefit of all cancer patients. It takes boldness, it takes bravery, and a lot of energy to keep challenging the status quo, but that's how we innovate. And it starts with courage, to build a team, to shoulder responsibility for putting someone's loved one through clinical trials, to spend hours in the lab testing and supporting bold ideas, then maintaining inspiration so the team shows up again and again, even after a fail, even after a loss. Advances in one cancer can and do impact others. Of course, there is always fear, but we cannot let that hold us back. It's important to have hope. So last year, Science and Technology Australian President Professor Mark Hutchinson gave the inaugural Hope Oration. Mark articulated what I've long understood as a medical oncologist who regularly sees patients with a terminal diagnosis, and what Richard has experienced as a patient facing a terminal diagnosis. That hope is an intrinsic and integral part of science. Hope empowers us to take action. It does not sit back and wait for things to happen. Innovation cannot succeed without hope. Even a glimmer of hope. Just one idea sets off a chain reaction of decisions for outcomes, for innovations. Because if not for hope, how else could we endure the loss, the setbacks, the long days in the lab, with the data, the hours at the microscope, or with patients deciding on drug therapies and clinical trials? As a patient-facing clinician, I absolutely love being able to now sit face-to-face with my melanoma patients and share how they will most likely survive. This just was not the case 10 years ago. I'm driven by hope for my neuro-oncology colleagues to have this experience with their glioblastoma patients, and eventually every single oncologist in the world. And critically, it is teamwork that underpins success. Teamwork fosters excellence, generates change in a much more productive way than the current reality. Teamwork must be at the forefront of our pathway to transform cancer research. We have made huge steps forward for some cancers, but we are not curing them all. And it's time the cancer research field, and indeed all medical research, thinks courageously to challenge the status quo. So, where am I on this journey? As you've now understood, I'm a medical oncologist, clinical trialist. I develop drugs, utilise drug therapy to tackle cancer. 50% of my 80 to 100 patients I see a week are on clinical trials. Finding drug cures is what I do. I work with the smallest of biotechs, the largest of drug companies. I review their drugs, help them design the trials, and they help me treat cancer. I never stop learning. Looking back, the last year has probably been the most challenging year of my life, but as fate would have it, it has also been the most productive academically. From my melanoma work, I've published extensively this year, and I'm proud to say, including 3 New England Journal papers in 2024, 2 of which I've led. I'm also currently on a mission to get another 2 drugs approved by Australia's Pharmaceutical Benefits Advisory Committee based on that phase 3 Nadina trial. The Netherlands already have the drugs funded, and Canada too, and they did not even participate in the clinical trial. Australia cannot wait this long to approve these drugs. The treatment is the same cost or less than the current standard and reduces the risk of recurrence or death by a near 70%. And quality of life is maintained. Where this is concerned, I am struggling to exercise patience. But what I have learned in my career is that there are many paths, not just one right way, to achieve an outcome or find a solution. If you're on your way down the river and it's blocked, look for another way. Look for a creek. There are many other rivers and creeks that head to the ocean. One of the best parts about this year is that Richard and I were named Australians of the Year for research in melanoma. It's an incredible honour, and the platform has given me an opportunity to share my story, to inspire people just like you to make this incredible nation we live in a better place. For this oration, I've focused on my area of expertise: medical oncology, drug development, clinical trials, how I got there, the learnings along the way. But to really make an impact, we must perform good science and design excellent clinical trials that answer a question in an area of unmet need. We must embed our research into our routine clinical practice, and we've got to think beyond N of 1. But better yet, we've got to understand the cause of disease and focus on prevention. So Richard and I have spent this time as Australians of the Year highlighting primary prevention of melanoma. We want to put ourselves out of a job by preventing it altogether. Let's drop the incidence of melanoma to near zero, and therefore no biopsies, no scans, no drugs. We have focused on sun safety in general, sun safety in sports, and stopping the glamorisation of tanning. A modern era disease will kill some of us. And we all know that a tan is simply our skin cells in trauma after UV exposure. This nation urgently needs to do better with prevention across the board and in our adolescents. Did you know that elite swimmers in Queensland have a 10 times higher risk of melanoma than the general population? In most cases, melanoma is preventable by being sun safe. Clothing, hat, glasses, shade, sunscreen. It needs to have equal status as other safety measures, seat belts, helmets. We need to make sun safety a part of everyday life. But until we get there, know the skin you're in. You know what I mean by that. If you see something changing, seek medical advice. And this year, we have also secured federal funding for a national pilot for melanoma screening in high-risk populations. Unfortunately, I'm not out of a job yet, and we have much more to do across melanoma and cancer more broadly: prevention, diagnosis, and treatments. So what do I think needs to happen to cure cancer? Clinical trial participation needs to be bolstered. In Australia, less than 6% of cancer patients were on a clinical trial, leaving the remaining 94% reliant on the standard treatment and the status quo. Now, that might be okay for some. Clinical trials need to answer an unmet clinical need. It needs to be designed in the right way, in the right population, and include rare cancers. For example, immunotherapy after radiotherapy and chemotherapy, and in patients receiving immunosuppression drugs for brain swelling, is unlikely to work. It never worked in melanoma. That's why we give immunotherapy up front in melanoma. Rare cancers like glioblastoma get knocked back for drug trials because they're a small market, often with a poor prognosis. That's not right. We need more cross-cancer fertilisation. What we learn from one, transfer to another. We must also embed research into our routine clinical care. It's part of our philosophy. Generate scientific data faster, change clinical practice for the better faster. Collectively, the global cancer community needs to be brave and bold. Rest assured, no matter what the barriers, there are and will always be some incredibly brave, clever, inspiring people challenging the status quo and transforming cancer research to make a difference for many. So as I continue on my journey, I want to leave you with this. The extraordinary is only achieved when we think big, embrace big risks, and think outside the box. And there is nothing better than an idea, especially one from outside the box. I look forward to taking your questions. Thank you.
Georgina, that was really extraordinary at so many levels, and we're very, very grateful for that. Also, just to acknowledge the many community members that we've got in the room, it's really important that you're here. Thank you for coming, because ultimately this is about you. The research is just a mechanism by which the health of Australians and indeed around the world can be improved. So I just want to acknowledge that. So we've got time for one or two questions, but Georgina will be around. So for those of you who'd like to meet with her and take questions subsequent, I'm sure you'd be okay with that. So there is a question online, I think. How are you progressing with the health warnings on caps?
I like that question, because what that person is really saying is caps aren't broad-brimmed and don't cover your neck and your ears and things. I think that's a good idea. It can be difficult to get things centralised and changed and you know, legislated or rules, but it's not impossible. And so that would be part of what we want to form, is a round table, and we've been trying to get this across with government this year on our platform, to look at aspects like glamorisation of tanning, laws, how can we change laws about social media, etc., or guidance. So that would be part of all of that discussion. Very important.
So do we have one or two questions in the room, or? Yeah. We've got one there and one here.
Thanks, Georgina. That was a wonderfully inspiring presentation. So my question's about your experiences with treating Richard, and what insights you've gained that would inform future clinical trials in glioblastoma, but also the past clinical trials. So I'm wondering what you think led to the failure of those clinical trials. Obviously, you think neoadjuvant is the way to go, but were there any other factors? Should patients be selected more carefully? Anything else?
Yeah, that's a great question. So anti-PD-1 alone was mainly the backbone that was used in those clinical trials, and I would never use that in the really bad to treat melanomas, but we've learnt that through our own experience. And so all the melanomas, there are melanomas where you look down the microscope, there's no immune cells whatsoever. They're really hard to treat. They're often very aggressive. I would never choose one drug. So that's one way we can cross-fertilise that these immune, not activated cancers, they will not respond to anti-PD-1 alone. So they're the trials that have been done so far to date. The other aspect is immunotherapies at its infancy. It's like the 1960s with chemotherapy. And people died from toxicity of chemotherapy back in the 1960s frequently, actually. We don't know how to. I think melanoma, we're leading the way. We've learnt through, just experience over 15 years, but we're not dosing it the right way. We're not managing it the right way, and we will refine that. But for example, chemotherapy needs to be dose dense. You have to do it at the cell cycle, so you have to give it every 2 to 3 weeks or whatever the regimen is, depending on the chemotherapy. But immunotherapy should not be dosed at a regular interval. Every human is different, so every human will need different intervals. Hang 10, give one dose. If you're seeing immune activation, a little bit of liver abnormality, grade 1, just hang 10. Do not go in with your second dose. Individualise your dosing frequency to the patient, and you will drop the toxicity rate. And then the last thing I was going to say about that is legacy drugs. So we were lucky in melanoma. We had no drugs. It really was a desert. It was terrible. My waiting room when I started in melanoma, I wouldn't see the same patient every 6 to 12 months. They'd die. Now we've got survivors. But we had no legacy drugs that we had to then add immunotherapy to. And so a lot of cancers where chemotherapy is active, they have these legacies and then have to try and work out how to fit in chemotherapy. And it's a little bit against how immunotherapy works. So now standard treatments are combining chemo and immuno, but I suspect in those cancers, you could box them and say, okay, some patients, I mean, let's take lung cancer, where they give all together, there are actually a large group of lung cancer patients for lung cancer that probably just need combo immuno done, finished quickly. Then there are some that get away with single immuno, and then the rest probably need chemo and the immuno is not doing anything for them. But because of legacy drugs, a lot of cancers are stuck, and that's where trying to push the envelope is needed.
Last question, Chris.
Georgina, it was an amazing presentation and really inspirational. But the question that I have for you is really around the clinical research and clinical care governance space. I mean, everyone that knows of Richard's stories is simply amazed at how quickly you acted, how quickly you got the teams together to do what you did for Richard and to start a treatment plan for him. But obviously you were sort of allowed to do that. And for many of us that work in the research space, there are a number of barriers, you know, in the way. And these are governance barriers that make it very difficult to kind of enact a plan like this. So I just wonder if you could comment on the research sort of governance landscape and how we could do things maybe quicker.
So first of all, I've got a lot of patients on phase 1 trials right now that probably would have died but aren't. They're doing really well. I don't talk about it. You don't talk about it until you've finished the work. I've also got a lot of patients on phase 1 where they are dying and then it's not working at all. I don't talk about it. So that's the first thing. We do our work. I'll be frank, it's distracting. I just want to get on with my work. So that's the first thing. Second thing, in Australia, we have an opportunity with the TGA for terminal illnesses to do things that are different if you've got a terminal illness. So you just need to submit to the TGA. And I had worked with the specific combo I used with Richard in a phase 1 trial in melanoma. I was very experienced with it. I had all the toxicity data. So you've got to have the toxicity data. Same with the personalised vaccine. I had all the toxicity data, the vehicle, etc. So that's why I could do that. But frankly, it's something that we should get done, get the trial, then we talk about it.
Please join with me in thanking Georgina for an extraordinary 2024 Chalmers Oration, for the science, for the passion, for the external focus, for so much. Thank you, Georgina.
Thank you. Thank you. And thank you, Professor John Chalmers and Alex. It's a real honour.